Welcome, everyone, and thank you for joining us for today's session on pioneering our understanding of the human brain. We conclude our clinical updates track and today's session with the accomplished Dr. Helen Bronte-Stewart, neurologist, neurophysiologist, and movement disorder specialist. Dr. Bronte-Stewart's team was the first in the United States to implant a sensing neurostimulator from which they could record brain signals directly and use the patient's own neural activity to drive the first closed-loop neurostimulation studies in Parkinson's disease. This work has led to the first multi-center international clinical trial in closed-loop deep brain stimulation for people with Parkinson's disease, and Dr. Bronte-Stewart will lead this. She's the John E. Cahill Family Professor in the Department of Neurology and Neurological Sciences at Stanford University. She's also the director of the Stanford Comprehensive Movement Disorder Center, the co-director of the Stanford Balance Center, and the division chief of movement disorders in the Department of Neurology and Neurological Sciences. For a complete bio on our speaker, please visit the Biography tab at the top of your screen. Dr. Bronte-Stewart's talk is entitled Closed-Loop Deep Brain Stimulation in Parkinson's Disease Using Patient-Specific Biomarkers and Therapeutic Window. My name is Dr. Roshana Kashemian, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. We would also like to extend our special thanks to the Brain Initiative Program Directors, and in particular, Dr. James Nat, for their efforts in organizing today's session. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting your questions during the presentation. Simply click on the Ask a Question box located at the far left of your screen and type your questions in the drop-down box. Our speaker will respond to your questions via email following the presentation. The presentation is educational and therefore provides free continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of your screen and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Helen Bronte-Stewart.
Thank you very much for that introduction. My disclosure is I serve on a Medtronic advisory board. In this talk, I will discuss our research into relevant neural and physiological signals that may be useful for closed-loop deep brain stimulation for specific Parkinson's disease motor symptoms, and how we have used a dual threshold control policy algorithm based on patient-specific therapeutic windows in the first feasibility studies of closed-loop deep brain stimulation for Parkinson's disease using a fully implanted neurostimulator. Deep brain stimulation is a continuous electrical stimulation of target regions of the brain, usually at high frequency, via chronically implanted electrodes. Currently, deep brain stimulation is what we call open loop, non-responsive, and continuous. And what does this mean? Well, open loop and non-responsive means that the stimulator cannot sense the brain activity it is modulating. It cannot respond to the patient's state, whether they're asleep or awake, whether they're at rest or active. It cannot respond to their dominant symptoms, such as tremor, or slowness of movement, or gait impairment, or freezing of gait. And very importantly, it cannot adapt itself to their current medication or dopamine levels. It's continuous. That means it's on all the time and cannot automatically change or optimize stimulation parameters or the location of stimulation based on patient's state or symptom. And it uses a one-size-fits-all stimulation parameters. So open-loop deep brain stimulation, as you know, has been very helpful, but at what cost? And what do we need to do to improve this and to develop closed-loop or responsive or demand deep brain stimulation for the brain? Well, we need to know relevant neural and or behavioral features of the disease state or symptom that will be reliable and specific control variables or drivers of a responsive or demand-based neurostimulation system. This is similar to the way the heart rate was initially used for the first demand-based cardiac pacemakers, which were designed once they could sense the heart rhythm to improve bradycardia without causing other arrhythmias. We also need to use an implanted concurrent sensing and stimulating neurostimulation system that works in freely moving people doing their normal activities of daily living. And we need to develop optimized, adaptive, and automated algorithms which are customized to the patient, their activity, state, and their symptom, and their medication levels. This next slide shows you the Activa PC+S Nexus D system developed by Medtronic, which is the first closed-loop deep brain stimulation system using a fully implanted concurrent sensing and stimulating neurostimulator, which we have used for Parkinson's disease. And I'm just going to take you through this slide. In the middle at the top, you can see a diagram of an implanted sensing neurostimulator, or the INS, which is implanted under the skin in the chest. Local field potentials, or signals from the brain, which are over on the right top panel, can be sensed and downloaded onto the neurostimulator. And in the top right panel, you can see a schematic of a Medtronic 3389 tetrode, or DBS lead, on a sagittal section implanted in the subthalamic nucleus. And we can record from this lead the recordings, the local field potentials, then can be downloaded from the INS onto a tablet and then onto a PC via a bidirectional Bluetooth-enabled conduit called Nexus D. On the PC, we can program the PC with the classifier algorithms, meaning what is sensed, and the control policy algorithms, meaning what is sent to the INS as the stimulation parameters, which is then sent back to the brain. Now, we can also use behavioral features of the disease, such as the measurement of tremor, which you can see over on the right in the bottom part of the panel, where a patient is wearing a smartwatch, a Bluetooth-enabled smartwatch, an IMU, and this work was done in collaboration with Jeff Herron and Howard Chisholm, the University of Washington, who developed a smartwatch that then was enabled to work with the Nexus D system. So we can using this system, we can develop either neural-based or kinematic, behavioral-based closed-loop deep brain stimulation. And now I'm going to show you our initial trial of kinematic closed-loop deep brain stimulation using a measurement of tremor, such as you saw in the previous slide. So we are measuring tremor from the smartwatch that's wrapped around the hand, but is also resting on the leg. And the other side, the contralateral side, the deep brain stimulation is on, so we don't have contamination from tremor from the other side of the body. In the top panel in A, you can see a red line, and there's a statement that says calibration and then closed-loop stimulation. During the calibration phase, all therapy is off, and we are measuring the intensity of the resting tremor. From this, we can develop our dual threshold algorithm, which is shown in the lower panel by the blue line and the magenta line going across the length of that panel. The blue line represents the upper threshold of tremor, and the control policy algorithm states that if the tremor goes above the blue line, then the stimulator is commanded to increase its intensity or voltage. If the tremor is in between the blue line and the magenta line, then the stimulator stays at the voltage it's at. And if the tremor goes below the magenta line, the stimulator decreases its voltage. What you can see from these two upper panels is the voltage is represented by the blue lines, and the tremor intensity on the lower panel is represented by the black lines. As the tremor increases above the blue line, you can see the voltage increases, and the system responded very nicely to tremor intensity. In the insert, shown by the red dashed arrow, you see a blown up time window of how we increased voltage and how we ramped it up and how we ramp it back down at half the rate that we ramp it back up to avoid tremor rebound. Using this kinematic closed-loop system, we showed that it was efficacious, meaning it had a statistically significant improvement in tremor, using less than 50% of the voltage that the stimulator would have used if it was on the open-loop model. And the actually stimulator was on roughly 51% of the time it would have been on in the open-loop mode. So this was quite a dramatic savings of energy. But I think one of the most interesting aspects of this is how everybody's tremor is slightly different. And you can see in this diagram, this is a different patient who had very intermittent tremor. And when the tremor occurred, the voltage went up, but then the tremor went away. And in the end, the closed-loop system was only on for 11% of the time that the open loop system would have been on. So this is our first example of patient-specific, customized, closed-loop deep brain stimulation, demonstrating that if you're using this demand-based brain pacemaker for tremor, you certainly can use this without having it to be on all the time for many patients with tremor. So going back to our diagram of our closed-loop system, as I told you before, we can now use the neural signals directly recorded from the brain and use those as the control variable to drive deep brain stimulation. But first, we have to understand what are the relevant neural control variables for disease like Parkinson's disease. And this has been known for many, many years and is shown here by a large study that we performed in the operating room after we had placed the deep brain stimulating leads in the subthalamic nucleus. And we showed that in 129 out of 130 STNs, using a peak detection algorithm, we could detect elevation in the power in the local field potential spectrum in the beta frequency ranges. What you see here over in panel B is you can see a schematic again of that subthalamic nucleus with the deep brain stimulating lead implanted in the nucleus. What you can see is these curves designated by the dark black line, the gray line, and the dashed line show the power spectral density diagram recording from each of the channel pairs from the tetrode. So the lowest one is actually from the most ventral pair, 0 to 1. The dashed line, which showed the greatest power, was recording from 1 to 2. And the middle line was recording from the most dorsal pair, which is 2 to 3. And you can see that there's quite a dramatic elevation in the beta frequency range of power, suggesting the presence of neuronal oscillations and excessive synchrony in this beta region, and this is called the beta rhythm. And in panel A, you can see time frequency spectrograms, which basically show the same thing. So would this be a relevant signal to use for closed-loop deep brain stimulation? Well, the beta rhythm in Parkinson's disease has been known about in human subjects and in animal models for over 18 to 19 years, and work done by Peter Brown's group in Oxford and groups in Europe and in Canada, where leads can be externalized, showed early on that this beta band is attenuated when patients take therapeutic doses of medication, showing that there was an inverse relationship between the beta band power and the mobility or medication state or therapeutic state. We then showed similarly that the other therapy that improves mobility in Parkinson's disease, notably deep brain stimulation, also attenuated this beta band. What you can see on the top panel here is the time frequency spectrogram, and the black bars represent randomized presentations of deep brain stimulation in different voltages. And over the group, you can see in the lower panel that there was a nice dose distribution attenuation of beta band power as we increased the voltage. What's interesting is this became significant as we got above voltages that basically become therapeutic. So now we have an inverse relationship between a neural control variable and the therapy, notably medication dose or deep brain stimulation voltage or current intensity. And can we use this now to control deep brain stimulation using a neural control variable? And so this slide shows you that in fact, we can, and that we have developed a dual threshold control policy algorithm now using beta band power instead of tremor intensity. And this is shown over in panel A. The blue line represents the dose distribution, the voltage distribution attenuation of beta band power that I showed you in the experimental model in the previous study. And the black lines of beta increase and beta decrease show you the dual thresholds. Now, as I told you, beta is inversely related to voltage. So the highest beta will be recorded when the DBS system is off, shown at beta rest on the y-axis. Then the upper beta threshold will be that recorded at a lower voltage, which I'll describe in a minute. And the lower threshold will be in between the beta power recorded at the lower voltage and that recorded at the highest voltage we want to go to for this patient, beyond which they might get side effects. So how do we determine that? Well, I showed you how we determined that using tremor intensity, And now we determine this using bradykinesia intensity. So in panel B, the upper panel shows you the raw beta power fluctuation. The middle panel shows you how voltage varies as we incrementally step or increase the voltage. And the lower panel shows you the angular velocity traces of a repetitive flexion extension movement demonstrating bradykinesia. What you can see is that as we increase the voltage, the bradykinesia improves, meaning the angular velocity increases. And from this, we can determine what is the therapeutic window for this patient, the lower voltage being that at which they get acceptable but not optimal improvement, and the upper voltage being that beyond which they get side effects. The lowest panel of this figure shows you how beta power varied over time using the dual threshold algorithm and how voltage responded appropriately. What was interesting was that determining the upper and lower voltage and therefore the beta thresholds was different for different people. So now I'm showing you something in a vertical distribution similar to what I showed you in a horizontal distribution in the previous slide. And if you focus on the time frequency spectrograms in the middle of this figure, you can see that we increased the voltage from 0 in the top panel to 100%, which was the upper voltage, in the lowest panel in 25% increments. And on the left side of your figure, you see the corresponding PSDs of the measured beta band power at rest, which is shown by the black line. Blue is 25%, red is 50%, pink is 75%, and green is 100%. And more importantly, we also show the change in beta power during movement, which is what the stimulator will be recording when the patient's moving around. Over on the right-hand panel, you can again see the flexion extension kinematics, and you can see that this patient began to get some improvement, although it wasn't dramatic, at 50% of the upper voltage. And so we might choose VL as 50% here, or even slightly higher than that, and VU obviously as 100%. And correspondingly over on the PSDs, the beta power, the beta threshold, upper threshold will be that red line, and the lower threshold will be in between the red and the green spectrum. But interestingly, in another patient, this might be quite different. And this patient, you can see that, first of all, they got better at lower voltages, meaning that at 50%, they were almost as good as they were at the VU. And similarly, the PSDs during movement showed not much decrement from the black to the blue, meaning 25% of the voltage, but really showed quite a dramatic attenuation after 50%. Again, guiding us to determine patient-specific thresholds using their therapeutic window, trying not to go too high in voltage and not to go too low. So the next slide shows you the results of a paper that was just published of the first feasibility study of neural closed-loop subthalamic nucleus deep brain stimulation in Parkinson's disease using a chronically implanted neurostimulator in patients who'd been on stimulation for a mean of 22 months, almost 2 years. And we demonstrated that this was safe and tolerable in both tremor-dominant and akinetic rigid patients. You can see in panel A and B that neural closed-loop DBS improved bradykinesia and tremor, bradykinesia in both the velocity of the movement and the frequency of movement. And in C and D, you can show that, in fact, there was cessation of tremor for 100% of the time in almost all subjects, and that the tremor level when recorded when it was present was very low. Notably, this was done using less than 57% of the total electrical energy delivered during open-loop deep brain stimulation. So I'll finish with our interest in studies, beginning studies in gait impairment and freezing of gait. And this slide shows you that we have developed several metrics for gait impairment and freezing of gait using, shown in panel A and C, a stepping in place task on dual force plates, where the patient is literally picking their feet up alternatingly at their own pace. And in a forward walking turning in barrier course, which is shown over in B and D, where the patient gets up from a chair, performs 2 ellipses around these 2 barriers, and then 2 figures of 8 through the barriers and around these tight corners, meanwhile having these barriers that are above eye level, causing them to go through narrow spaces. What we have shown is that the stepping in place task and the turning in barrier course shown here in CND and ENF respectively reliably elicit gait impairment and freezing of gait, which however is not very well elicited when the patient is just doing normal forward walking in the clinic, as is well known. These traces are those of the angular velocity from an IMU placed on the lower leg of patients, and you can see the red and the black indicate each leg. And during forward walking, there's nice alternating strides, which do not show any impairment. But once they do stepping in place, you can see with the freezer that there is a time when they don't get their feet off the force plates. So they're freezing and their gait becomes more arrhythmic. Similarly, during the turning and barrier course, the gait in the freezer patient tends to break down, and they tend to have periods where they can't move forward at all. The gait becomes very arrhythmic, whereas in the non-freezer, their gait is fairly normal, and they do the normal slowing as they go around the corners. That is not a freezing event. We showed that most freezing events occurred when going around these corners or through narrow openings in the turning and barrier course, and this was similar to what the patients told us when they were in their home environment. So we've got kinematic or kinetic signals or behavioral signals of gait. What about neural signals? Well, here you see in this figure concurrent neural and kinematic recordings of a patient performing 100 seconds of repetitive stepping in place. I'll take your attention to the lowest panel to start with in A, where you can see the kinetic traces of a patient performing repetitive stepping in place. This we initially published in 2011 with Nantel as the first author. What we do is we normalize the force to their body weight. So you can see on the y-axis, we go from 0 to 100. At 100% of body weight, all the weight is therefore on that one leg, and therefore the other leg should be at 0%. For normal gait, there should be a nice alternating sinusoids out of phase with each other, so that when one leg is at 100%, the other leg is 0, and that changes as they step. What you can see is that this patient starts off very well with nice alternating stepping, but then this stepping breaks down, and when the traces don't reach 100 and 0, it means that the legs and the feet are not coming off the force plate, which is a very reliable way of detecting a freezing episode, which is shown by the letters FE above the freezing episodes. Using concurrent and synchronized neural recordings from the subthalamic nuclei, we can also look and see what is happening to this beta rhythm. Now, the second and fourth panel are the time frequency spectrograms from each STN. Above that, what we've done is we've taken the movement band, which is shown in the panel B, as the frequency band outlined by the dashed lines of the blue PSD, which is the one we recorded during stepping in place. We take that frequency band and we filter the raw signal to that band. We then rectify that signal and square it to determine how that band behaves over time. What you can see here are the dynamics of what we call the movement band over time during this task. During the normal stepping, there are these blue rectangles, which shows that there are fluctuations in the power in this band that have a fairly short duration. And these have been determined in animal models by Anne Grabiel's group, and now in Parkinson's disease by our group and by Peter Brown's group, as physiological or normal beta bursts, what we would expect to see in the brain. However, as you can see, as the gait is breaking down, we're also seeing increased power and increased duration of these beta fluctuations or beta bursts, which are thought to be pathological and related to the Parkinsonian state. And this paper was recently published showing that beta band bursts are indeed pathological in gait. To demonstrate this, this is a three-dimensional rectified time frequency spectrogram during gait. And what we've done is we've taken 2 Hertz bands and we've done the same rectification and squaring of the signal so that we can really look in a three-dimensional fashion at how the signal is varying over time. We also show the kinetic trace of repetitive stepping in place, synchronized in time with the neural recording. The upper 3D spec here is that of a non-freezer who performed the repetitive stepping in place very well, and the lower panel is that of a freezer, in which you can see again the stepping in place starting off well and then breaking down over time. And what were the results? Well, we found that in that movement band, power was higher and burst durations were longer in freezers versus non-freezers during normal gait. So not when freezing, but actually during what looked like normal stepping in place. We also showed that among the freezers themselves, movement band burst durations were longer during the periods of freezing of gait in freezers than compared to during a normal gait. When we applied lower frequency, 60 Hertz, which is known to be helpful for some people in freezing of gait, and higher frequency, 140 Hertz DBS, both improved the behavior, the freezing of gait, and attenuated beta power and shortened the movement band burst durations in the freezers. What was very interesting was that both frequencies of deep brain stimulation left unchanged the normal gait and the shorter burst durations in the non-freezers. And this was our first glimpse to suggest that, in fact, during gait, the effect of deep brain stimulation is basically to lift off that pathological network, hopefully leaving the physiological network intact. So how can we finally take this to closed-loop deep brain stimulation? Well, this final slide shows our early trials of closed-loop deep brain stimulation for gait using bilateral, laterally independent control variables and control policy algorithms. In this slide, we are using the Nexus D system, in which we're basing this off beta band power. But as we move to the new system, which is called the Summit RC+S system, under a Brain Initiative UH3 grant, we will be now able to do this based on the actual dynamics of the beta band rather than just power. In panel A, you can see a patient performing repetitive stepping in place off all therapy, and you can see that the normal stepping in place starts breaking down, becoming irregular, and finally landing up in periods of freezing of gait where the feet do not get off the force plates. Under open loop deep brain stimulation, the patient does much better, but you can still see that there is some arrhythmicity and some freezing episodes recorded during stepping in place. Panel C shows closed loop deep brain stimulation using the neural control variable, and in the top two panels you can see how the voltage changed in the two hemispheres, and in the lower panel you can see that the stepping in place was now performed very smoothly and without freezing episodes, giving us a very hopeful sign that in fact closed-loop deep brain stimulation may be very useful for gait impairment and freezing of gait, which are currently quite difficult to treat. Finally, I'd like to thank all the current and former members of my lab who've contributed to this work over the last 4 to 5 years. Our collaborators at Stanford, our neurosurgeons, Scott Delp, bioengineer, our collaborators at the University of Washington, Cameron McIntyre, and especially our engineer collaborators at Medtronic. But most importantly, I'd like to thank the people with Parkinson's disease who have volunteered many hours of their time and energy, and without whom none of this research would be possible. And we are very grateful for the support of this research from the groups listed below. Thank you.
Thank you, Dr. Bronte-Stewart, for presenting that pioneering talk, which will help those who are suffering from Parkinson's disease. As a reminder, our speaker will follow up with any questions about her presentation by email. This concludes the Pioneering Our Understanding of the Human Brain session and day 1 of Neuroscience 2019. Today's presentations will remain for on-demand viewing through June 2019. We've got more excellent talks by outstanding researchers lined up for tomorrow, day 2 of our Neuroscience 2019 conference. Tomorrow, we'll explore perspectives on neuropsychiatric disorders and look at the emerging role of the immune system in brain function and behavioral processes. See you tomorrow.